to introduce Amber Benezra. I'm so glad you can join us. I'm so glad I'm finally getting to be in person in real life, not in Zoom land. She got a bachelor's of arts from Carnegie Mellon in 1996 and also did a master's at the New School for Social Research in 2000. But I think she probably began most of her work that we'll be more familiar with after 2007, when she got a master's at the New School for Social Research, sociocultural anthropology. And she got a PhD in 2014 in the same topic. I think we know, or at least I know, a lot of Amber's work from time she spent at the International Center for Diarrheal Research in Bangladesh, or the ICDDRB, which several of us in this room have involvement in projects with them, so that's great. And she was also a visiting researcher for Jeff Gordon's lab at Washington University, which I think is just awesome. And so I have been a fangirl of Amber's for a long time because she has an awesome paper called Anthropology Microbes that I assigned to all of my Anth 414 students. So many of you in this room have read that paper because I think it's an awesome way to integrate anthropology, bioethics, and microbiome sciences. So, you know, it's one of these things where interdisciplinary research is so important here at Penn State. I think Amber's work is a really good model of how far you can extend that, and including extensions of that outside of the hard sciences. And if you weren't aware, she has a book that's just come out, which is super exciting. I have my copy here to get it signed. It's called Gut Anthro, an experiment in thinking with microbes. So with that, I want to present to you with our nice little plaque to say thank you very much for visiting us here at Penn State.
Thank you so much.
And otherwise, welcome, Amber. We look forward to it.
Thank you. It's my first plaque. You guys, I'm so excited. I'm really excited to be here. As Laura said, I have this book. So I have 2 children, but this book was more work than making my action. It took me 6 years to write it. I am selling them, which is just like a sad commentary on academic writing. You know, when I got a book contract, people were like, yeah, book contract, you got a good advance, like you're gonna make a lot of royalties. I was like, no, that's not how academia works. I will never see a dime from this, but I do have copies for sale. So that's hawking my book. That's part of that. Thank you to Laura. Thank you to the One Health Microbiome Center for inviting me. I am a sociocultural anthropologist. So what I'm going to talk about today is probably a little different than most of the talks you hear. But I think you'll like it anyway. I'm super excited to not have to explain to you what the microbiome is. Usually that's how I have to start most of my talks. I'm like psyched that I don't have to. But anthropologists typically read. I'm like trying to gauge the crowd. I don't know if I should read my paper or not, so I might like mix it up a little bit, do a little bit of reading and a little bit of not reading, but we'll see how it goes. But I'm really happy to be here, so thank you. Dhaka, Bangladesh looks lovely in the earliest hours of the day. When the sun rises over the bamboo scaffolding of half-finished high-rises, the people are quiet and birds and insects fill the sky. The morning light is beautifully tinged with exhaust, pollution, and dust. Within minutes, it becomes intense, bright, and hot. The sun starts early. In Mirpur, one of the most densely populated localities in the most densely populated place on Earth, mothers rise in one-room shacks. They chase mice from cooking pots, boil water, and prepare food for the day. 8,000 miles and 11 hours later in St. Louis, Missouri, scientists will arrive at the Gordon Lab, sacrifice mice, segment intestines, and prepare cells for sequencing. Microbes in the guts of these mice are from the guts of Bengali babies. They traveled across the world in test tubes, and fecal samples collected by mothers and packed in big coolers in the stifling Dhaka sun. Microbes traveled by airplane on flights that cost more than the yearly income of most Bangladeshi families. They arrive at the Center for Genome Sciences and Systems Biology at Washington University, a gleaming multi-million-dollar facility with several sequencers, a mass spectrometer, and a computer cluster capable of analyzing trillions of reads. These microbes will be injected down the throats of black 6 mice used for diet experiments on the Bangladeshi microbiome. What I want to say here is more than a predictable cross-cultural comparison between a globally southern, resource-poor community and a globally northern scientific research laboratory. These spaces are more than geography, more than poverty, more than notions of power, histories of colonization, more than practices of living and knowing. They don't just overlap the boundaries of the body, Between the Gordon Lab and Mirpur, the social and biological become equally important to understanding human microbial relationships. So based on a collaborative partnership with scientists working on the microbiome, my research follows commensal microbes in the human gut as objects around which scientific and anthropological imperatives are emerging. I conducted ethnographic fieldwork at the Gordon Lab at WashU, where scientists were studying the interrelationships between diet and microbial community structure and function, and whether differences in human gut microbial ecology affect predisposition to malnutrition in children. So there was a maternal child health study called the Microbiome Discovery Project. It was funded by the Gates Foundation and part of the Malady Networks, which is malnutrition and enteric disease. So they have a bunch of global health sites. Bangladesh was just one of them. They have them in Peru, Malawi, South Africa, all over the world. So the Gates Foundation funded that study that was going through the Gordon Lab. I also worked at the lab's field site in Dhaka, where Bangladeshi mothers and children were enrolled in this study. There I worked with scientists and researchers at the International Center for Diarrheal Disease Research, which is known locally as ICDDRB, also known locally sometimes as the cholera hospital, because in Dhaka they have something that they call cholera season, where they literally fill all their beds and then fill the parking lot with triage tents. The Gordon Lab was attempting to develop a translational research pipeline in order to alter the microbiota of those suffering from metabolic disorders and nutritional deficiencies. I followed microbes to homes in Dhaka, where microbes were both conceptually non-existent and practically a matter of life and death. As lab members studied the relationships between gut microbes and malnutrition, I explored how an anthropologist could become a member of a scientific team and contribute ethnographic data while asking biological scientists to make space in their research design, experimentation, and implementation for a social science expert. So thinking about ethical and social and legal implications, not as sort of an afterthought as the thing you have to like tack onto your NSF grant because you have to meet your ethical guidelines, but upstream in experimental design, thinking about how to do this work from the start with these social science practices in mind. This is the paper that Laura is referring to. So in our perspective piece in PNAS, Jeff Gordon, Joe DiStefano, and I created what we call the Anthropology of Microbes. And we really believed that methods for understanding the microbial world provided an opportunity to reevaluate both how we think about human biological and cultural diversity, and really necessitates a collaboration between different kinds of experts. So thinking about how the natural and social sciences might use studies of the microbiome to jointly engage pressing public health problems like malnutrition. So if we want to approach and solve and work on these problems, how do we do that together? So here's some questions you probably have at this point in the talk. First one, what do anthropologists do? Some of you may be biological anthropologists. So anthropology typically has a four-field division. There are four subfields. One is biological anthropology, which we consider the hard scientists, right? Biological anthropologists are scientists. Sociocultural anthropologists have nothing to do with biological anthropologists. Sadly, there's a very big rift in our discipline and we don't really like each other or work together very often, which is not great. There are also archaeologists. So we always think about Indiana Jones when they think about anthropologists. And there are also linguistic anthropologists, which doesn't exist anymore too much, but those are the four fields. But biological and sociocultural are the main anthropological subfields. So sociocultural anthropologists study the complex cultural lives of people. We study their decisions, their social practices, and their relationships. To be completely honest, the history of anthropology is extremely colonial and racist. Anthropology was founded in some very white Euro-Americans going to study foreign lands and learn about like rituals and kinship structures. We spent the last 50 years trying to sort of undo that terrible beginning of our field. And so that's not what anthropology is about anymore, but sort of our origins. But we use the methodology of ethnography to ask questions about people in the world. We do what we call fieldwork, ethnographic fieldwork. So that's me in the field. The ethno part of ethnography is observing and talking to people who are going about their everyday lives as they experience the commonplace and the extraordinary. And then the graphy part is when we take all that information, we analyze it and write about it. Our work is primarily qualitative. We do not produce clean, noise-free data, and our sample sizes are very tiny. So for instance, my N is 9. I talked to 9 families during my fieldwork in Bangladesh. So the next question you might have is, what does anthropology have to do with microbiomes? So as an anthropologist, what I love about the microbiome is that it brings together the social intimacies of our lives with our biological selves in ways that show us that these two things are inextricably entangled. So microbial populations are affected by how and where we're born, what food we eat, who we live with, who we love, if we have a dog that licks the inside of our mouth every day, right? If we wear shoes to the bathroom. And those microbes are digesting our food, training our immune systems, interacting with our states of health and illness, moods, and behaviors. So we know that metagenomic technologies are revealing the impact microbial communities have on our human health and changing ideas about biology. In turn, social scientists like me are beginning to have microbial concerns as we start to think about how poverty, race, lack of access to healthcare and food all also shape our microbial populations. So how can anthropologists and human microbial ecologists, really it's a typo, should say ecologists, work together? So sociocultural anthropologists do not like to make generalized conclusions about cultures, societies, or groups of people. I would never say I know what happened in all Bangladeshi families because I worked there. I know what happened with those 9 families that I worked with, right? We're pretty loath to make any generalizations. But ethnography does provide insights into what life is actually like for people on the ground, giving important details that might not be visible in something like microbial genomics. It can shed light on the transnational and local realities of people embedded in global scientific research and health programs. It can show why health interventions don't work, how sometimes they're not practical or implementable in real life. In my subsequent research, I've continued to develop this idea of anthropology of microbes, drawing together the collaborative capacities of ethnography to actively contribute to scientific projects, partnering with scientists to develop a more holistic, analytical, and methodological framework for understanding the embodiments of biology within social dynamics. So last question is, isn't transdisciplinary collaboration hard? The answer is hell yes. It is very, 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 very difficult. So, interdisciplinarity is a buzzword. Everyone loves to talk about it. Everyone wants to do it. It sounds really great. But when you're applying for a job, there are no faculty tenure track jobs in interdisciplinarity, right? There are very few interdisciplinarity journals. There are no grants in interdisciplinary. So institutionally, those things still have really rigid disciplinary lines, and we have to follow them in order to have any kind of professional success or progress, right? So it's great to call yourself an interdisciplinarian, but where does that actually get you, right? Also, even if both parties in the partnership, for instance, Chef Gordon and I were totally in it. We were like 100% committed to our collaboration. We came to the table with open minds, willingness, and generosity, and we still had a really tough go of it, right? We're all trained in our own languages and our own methods, and those things don't really mix, right? When Jeff and I went to write this paper together, we didn't structure sentences the same way, right? We didn't make arguments the same way. We argued about how to just lay out a paragraph, let alone what information to convey, right? None of us are taught to do that sort of cross-disciplinary talk. So that stuff is really hard. Even if you like the person a lot, even if you're totally committed to making collaboration work, that stuff is tough. So that's something that I think is very worthwhile to figuring out, but no one really tells you how to do it, right? All of our disciplines have a particular language. We conduct research a certain way, and we don't learn how to talk to each other. So I worked collaboratively with the Gordon Lab for about 7 years. And for 14 months, I was actually in the lab conducting my independent research and also serving as an anthropologist in the employ of the lab. I was thinking about how accounting for microbes changes anthropology and how anthropology can collaborate with scientists accounting for microbes. So a lot of the Gordon Lab experiments when I was there, which was 2009 through 2011, we're looking at the gut microbes of babies in Bangladesh. They also were doing work in Malawi at the time, who are suffering from malnutrition, trying to figure out the connections between bacteria, food, and illness. So they enrolled mothers and babies in this study. They took lots and lots and lots of samples of poop. and also blood, urine, and other things. They gave these mothers free healthcare and medicine. So I went to the field site. I spoke to people, the Bengali field researchers, the mothers enrolled in the study, and tried to think about the structural issues affecting their lives, right? Poverty, poor sanitation and infrastructure. And then on the other end of the research network, I also talked to the Gordon Lab, PhDs, postdocs, lab techs, thinking about asking them about how they thought about the work they did, and their own relationships to their microbes, which was a pretty fruitless endeavor, honestly. Every scientist, microbiome scientist I've ever talked to, when I ask them how they think about their own microbes, they're like, take less antibiotics and eat more fiber. And that's the only prescriptive I can give you about microbiome. I'm like, all right, fair enough. So now when people ask me, because they think I'm a microbiome expert, what they should do, I'm like, those are my 2 words of advice for you. So I asked then, and I continue to ask, what is at stake in these human microbe relationships? How do we address bioethical issues? Why are certain bodies, especially those from historically vulnerable populations, the focus of microbiota sampling? What happens when scientists talk uncritically about people of different races having different microbiomes? How can we make sure that the study subjects are heard and also benefit from the research? How does our genomic intertwining with our microbial partners change how we understand biological relatedness? What does it mean to define our states of health through our microbial selves? So when I was thinking about talking to this group, I realized that social and biological scientists may not even start with the same idea of what social determinants are, or even what health is, right? So for human microbial ecologists, the health that's important when studying the microbiome might consist of diet, geography, genes, mode of birth, and vaccines. So these are all things you might take into account when you're thinking about microbial populations in the gut. As an anthropologist thinking about mothers and babies in Dhaka, all of those things matter, along with important influences like gender and class and ethnicity, working in environmental conditions, in garment factories in Dhaka, for instance, the violence of a colonial national history, corrupt or non-existent governmental systems of sanitation and water. So when we start to pile up all these concepts of health on top of each other, the picture becomes really messy. It's very confusing to parse out what's what there, right? But it also becomes more detailed, more complete, and eventually, hopefully, more precise. This is why microbiome science and anthropology need one another. You start with this picture, you're like, cool, that makes a lot of sense. Let's just do that, right? Science is like pretty excited about that picture. This one is like really complicated and hard, but it doesn't mean we shouldn't do it. So while I was in the Gordon Lab, at least 6 lab members were working specifically on gut microbes and malnutrition. 3 were dealing directly with the samples from Dhaka. My research focused on 2 projects, one analyzing the Dhaka samples and the other conducting diet experiments using what they called Bangladeshi food and germ-free mice humanized with microbiota from Bangladeshi donors. I was specifically interested in these studies because they analyzed microbial material from babies in a specific geographic locale with a specifically defined health and nutritional status. These studies involved human beings I would be able to talk to, that I could travel to and observe. I wanted to find out where the feces came from, what the conditions were in the place they were produced and for the people producing them. So I will say at this point, in recent years, I've become very, very aware of the pictures I've taken of my Bangladeshi subjects. You will notice there are no pictures of lab members in my slideshow. That is a problem and something that I am really grappling with, because I realize in hindsight, it was very easy for me to make Bangladeshi people subjects, right? They were naturally research subjects, but there's so much class and race privilege on my part that allows me to do that. I felt really weird about asking Jeff Gordon to pose next to his like mass spectrometer. Like I never would do that, right? I didn't ask anyone in the lab to pose for pictures, and that is a problem. So this may be the last time I use these pictures in my slideshow because it's problematic. And while I think it's really important, especially for the Gordon Lab scientists, to see the humans, right? They just got a shit ton of actual shit from Bangladesh, right? but they never thought about the human people. So I think that it's really important to see faces on human beings and think about the broad and complex lives of scientific subjects. But portraying people in this way is not feel good, and it's not necessarily right. So I still haven't figured it out, but I want to make that comment. Right. So I was interested in how Bangladeshi diets were represented in mouse experiments. This is this huge industrial mixer, so they make all their own mouse chow, and also like for the diet experience, they called it chow baka. So for instance, so what they would do to make the Bangladeshi diet, so humanized germ-free mice with these samples from these babies, and then they would go, you know, the scientists would go to Whole Foods in St. Louis and get spinach and lentils and rice and make a, you know, approximate Bangladeshi diet. So I was interested in that because obviously so many things about how the rice is grown in Bangladesh, right? Like pesticides, all kinds of things also have an impact. But also Bangladeshi food is full of chilies and garlic and all kinds of things that would kill mice. So they don't put that in. So, I mean, the Corn Lab scientists knew all of this and they're like, this is the best we can do, right? This is an approximate diet intervention, but it, for me, as a non-scientist, that was a really curious thing to think about. So I followed the fecal samples as they traveled, as they were made by human and microbial bodies, and as they arrived in the lab and were translated and analyzed. Oh, there's some chilies being dried. Often our host families and the people who were feeding us told us that they were feeding me baby food because like, they're like, as a white person, you can't handle Bangladeshi food. And I was like, fair enough. Okay, thank you. But no, lots and lots of chilies. So the birth cohort in the report consisted of children whose mothers were enrolled while pregnant. These mothers received extensive prenatal care, education on healthy eating and breastfeeding, and they gave birth at health centers rather than at home, which is actually the local convention. So the case-controlled children were given macro and micronutrient supplements as well as therapeutic foods, and all the families received healthcare in exchange for their participation. So even this is somewhere where I can add in sort of anthropological anecdote. We're like healthcare, that's great, that's a good benefit, right? But that care was not value-free. So what I found out through my ethnographic research was that some of them, so they really didn't prescribe a lot of antibiotics in this study. Obviously, they're studying microbiome. They don't want to over-prescribe antibiotics. So the study doctors wouldn't really prescribe antibiotics as much as the mothers were used to. And the mothers didn't like that. They knew brand names of antibiotics, and they thought to them, they said actually to me, this healthcare is shit because they're not giving us the medicine we need. It's not good care because it's free. So they would take antibiotics on the sly and not report it to the field research assistants. So that is an interesting thing that happens on the ground, right? And obviously it affects your data and your study results, but also you have to think about what people do in their lives, what they think, what's meaningful to them, right? And how that's going to affect the scientific work. So each family was visited regularly by field research assistants. Here's one taking some probably diet recall here. They collected socioeconomic, surveillance, and food recall data. Sample collection and surveillance of cohort children began within 72 hours of birth. So they monitored the mothers prenatally, and as soon as babies were born, they sampled their meconium. So the first stool of the baby's lives is the first sample they submit into the study. Blood, urine, and fecal samples were collected pretty much daily, sent to ICD-DRB, and then sent to the Gordon Lab for metagenomic analysis. So there's this is what the mothers would sample with, and they would put them in picnic coolers. So I often thought this also was a very interesting thing for me to think about. So the Gordon Lab is very well funded. They have negative 80-degree freezers that have alarms on them in case the power ever goes out. But this is where the samples started, right? So the mothers were taking the samples in these very contaminated conditions. They were being put in picnic coolers and then into refrigerators that were regular fridges in which the power would probably go out 5 times a day. So when we think about, they were like, there's such a carefulness about the samples once they got to St. Louis, but where they originated, there's quite a disconnect between those things, right? Which was a very interesting thing for me to think about. So The birth cohort in Mirpur during my time in Dhaka, 3 Bengali field research assistants introduced me to families in Mirpur enrolled in the study. Children in their community suffered from extraordinary bouts of diarrheal illness. So malnutrition in these communities is not because of food scarcity, but because of enteric disease. There is so much diarrheal illness, your body just can't hold on to any nutrition. So one doctor told me there was one child that had 300 episodes of diarrhea in their first 3 years of life. So malnutrition was often the result. The Gordon Lab was analyzing the microbial genomes of fecal samples from these babies to see what microbes they had and what the microbes were doing. I served as the lab ethnographer. I gathered information on lifestyles, diets, and practices of everyday life. I want to give you a little bit of an ethnographic snapshot to think about how we can start to overlap the scientific and social concepts of health, how to use different kinds of expertise and data to do the most ethical and effective science possible. The solution isn't just to sample and study more diverse populations to have wider representation. That's important, but it's also to work with social science experts who are able to ask different kinds of questions, who can think carefully about who's going to benefit from the science, where does the power lie, and what are the social, political, economic conditions of life for the people we're studying. It's crucial to hear the voices of those people too. So this is Rimi and Kanta. Kanta is the baby. She does not like eating. She's 18 months old and quick on bare feet out the open door of the nutrition center into the hot Dhaka street 3 times before her mother Rimi catches her. Typically gentle and affectionate with her daughter, Rimi holds Kanta firmly in the green light of the nutrition center under illustrated posters teaching about nutritious food and hand washing. Kanta cries loudly as Rimi tries to spoon cereal into her mouth. Quote, she hates pushti packets, but she doesn't like any food, so we force her to eat, end quote. Pushti packets are locally produced, ready-to-eat therapeutic foods, macronutrient meals containing rice powder, lentil powder, molasses, and soybean oil. So if you've ever heard of pushti packets, plumpy nut, right, is like a ready-to-use therapeutic food that's globally distributed and mostly in malnutrition crisis cases. What the Bangladeshi researchers found out is that, so plumpy nut is a peanut butter paste that has a macronutrient profile that they give to kids, usually in disasters, right, or where there's like really severe malnutrition. What the Bangladeshi researchers found was that Bangladeshi kids don't like peanut butter. They're like, what the hell is this? We don't like this consistency of it. We don't like the taste of it. It's not part of their normal diet. So this is another example of like, you know, very westernized idea that like all kids like peanut butter. So they made a local, like from local ingredients that had the same sort of nutrition profile, therapeutic food. The Bangla word pushti translates to nutrition in English. Although half of all infants in Bangladesh weigh less than 5 pounds at birth, it is when children get very sick that families seek help from ICDDRB, which has an influential historical presence in Mirpur. After local health workers diagnosed Kanta as malnourished, Rimi enrolled her in the microbiome discovery study. Quote, I thought my baby had a vitamin deficiency, and most importantly, I wanted medical care for her. It's very good for my baby because she is underweight, and now she is in good health. I don't mind giving feces and blood samples. They can use them to find if my baby has any diseases. They will know about it and will treat it, end quote. So Kanta and Rimi live in Block D of Mirpur 11 in a 3-room house with tin walls. In one room, Kanta sleeps with her mother and father. In the other, her grandmother, grandfather, and their 9-year-old son. The 3rd room is occupied by a renter and her young daughter. 8 people inhabit this 20 by 24 foot space, sharing the attached bathroom and narrow kitchen with 2 gas burners, a supply water pump, and a pit latrine. Daily, government and NGO tankers truck water into communities and fill underground tanks, which are connected to above ground pumps. Drinking water in Dhaka is notoriously and extraordinarily contaminated with sewage, agrochemicals, oil spillage, solid waste, and untreated industrial effluent. While boiling can eliminate some contaminants, it does not rid it of high levels of arsenic and other heavy metals. So this is another sort of ethnographic example where while I was in Dhaka, there was a huge water boiling campaign, a public health water boiling campaign. They were telling everyone you have to boil your water before you drink it. It's not healthy to drink. It's going to make you sick. So everyone got this message. When I went and was with the families, I noticed that no one boiled their water. I was like, what's going on, y'all? Like, you heard this water boiling thing. It's not healthy for you. It's got a bunch of shit in it. You really need to boil your water. What I found out is, so they have these, so sometimes 5 families will share these 2 burner stoves in their kitchen. And the gas service is extremely unreliable. Most of the time it goes off by 9 a.m. in the morning. So mothers were making these very deliberate decisions to cook food for the whole day for their families. Instead of, they're like, I'm not spending an hour boiling a huge pot of water. That's a waste of time and gas. So in order for my family to have dinner tonight, I need to have food cooked now because I won't be able to cook any other time of day. So they're making these prioritized decisions practically about what they can do. So we see again how interventions fall short or just aren't implementable when it doesn't make sense for people. They're not going to spend hours boiling a pot of water while they need to actually cook food, right? Even though they know very well the water is not good to drink. So in Kanta's house, there are no doors, no windows, and no shoes. On her street, there are chickens and ducks underfoot and an open drain in front of every house. Remy cooks food for the whole day before the gas service goes off at 9, preparing huge pots of rice, dal, spinach, and potato mash, and negotiating time at the stove with 5 other families who share it. So the way in which Kanta's microbiome, her microbial self, comes into being is dependent on its social as well as its biological location. Along with birth, breastfeeding, and genes, it's shaped by local practices surrounding water, chickens, and bare feet. The microbial populations in Kanta's gut are specific to the dilemmas that these bring into being. But how do we account for these social and environmental factors? Malnutrition is a complexly historical, context-dependent, socio-biological state informed by microbes. The Gordon Lab members were very mindful of how many potential interacting explanatory variables affect malnutrition. Yet ways to sort of incorporate or correlate the ethnographic data into the data analysis of the microbial genomes has yet to be devised. I've been doing this work for 14 years now, and I still don't have an answer for that necessarily. But yeah, let me keep trying. Since gut microbes are studied and understood through their collective genomes and metabolic processes, studies of the human malnutrition vis-a-vis microbiomes are at risk for resulting in public health interventions that only focus on fixing the microbiome. So what's happening with the Gordon Lab work now, since I've not been in the lab anymore, is having microbiota-directed therapeutics, probiotics, microbiota-directed foods that will treat what they call the malnourished microbiome. But my point as an anthropologist is that we can't just fix the microbiome, right? What about all of those other ways of living that are also factors in people's lives and will continue to be? What are the ways in which we can account for biology and social inequalities, like poor infrastructure and poverty that produce human and microbial selves? What if we consider the social simultaneously and inextricable from the biological? Not as metadata, but as crucial interacting data. As microbiome science endeavors to use computationally derived data to develop probiotics as treatments for the broken microbiota, can collaborating social scientists make sure these interventions don't circumvent the complex and confusing social conditions that people have to survive every day? On my last day in Dhaka, I asked the FRA supervisor, Adila, who's in the middle there, if she had any other thoughts for me to take back to my lab colleagues. She thought for a moment and said, quote, "Mothers are the best people to know what kind of illness their child has and what kind of treatment they require. I've learned a lot from working on this study. When I see a malnourished child, now I think, what's going on with that child? What are the factors in his life causing this? The scientists have to come here to see it. There are little things I can express and things I can't unless they come here. They don't know what it's like. I can only tell you my opinion, and it's too hard to explain the conditions here without seeing it for yourself." Adila doesn't just mean the state of things in Mirpur must be seen to be believed. She's saying that both human and microbial bodies are materially affected by the social circumstances of everyday life in Mirpur. She knows that ICDDRB and Gordon Lab scientists receive hundreds of thousands of biological samples. She and her team collect, label, and pack them. She also knows what prescriptives about breastfeeding and antibiotic use circulate from above after the samples are analyzed. So samples come from Bangladesh, go to the lab, are analyzed, and then diet prescriptions, hand washing, and breastfeeding prescriptions come out of the lab back to Dr. Adila mentions to me more than once that these mothers don't care about microbes. That getting free healthcare for their children, keeping them fed and without diarrhea is all that matters. So this is the picture that was all over ICDDRB. So all of the mothers in the study were only taught about pathogenic microbes. They were only taught about microbes as disease-causing. Never talked about commensals or good microbiomes, or thinking about microbes that are necessary for health. They just weren't even introduced to that concept. They were only taught about hand washing and hygiene, and microbes as bad, essentially, which is, I don't know how to resolve that disconnect between what the science is trying to accomplish and what people understand about the science, from which their bodies are producing the raw material for the science, right? There's an interesting disconnect there that I'm not sure how to resolve. I could keep talking about race and microbiome, but I don't know that we have time to do that. There's a whole other thing, which is kind of where my future research is going, thinking about how to create an anti-racist future for microbiome science. But maybe I'll just leave that for now because I'd rather stop talking and hear from you. Unless someone's dying to hear about it. Thank you, that's okay.
Yeah. I kind of want to hear about race. How many of you are curious about the race?
All right, I'll go through it quickly. Sorry, all right, so a lot of my new work has been working with a lot of junior scientists. Something that I'm really excited about the future of microbiology is that junior scientists have core investments in things like social justice, and it's important to how they want to do science in the future. So this is something that I've been dealing with in a couple of papers in the last couple of years and thinking through it. So we can all agree race is a social construct. I think that's like a general thing. Everyone's like, yes, I'm on board with that, right? So what does that actually mean though? And how does that play out in scientific work? So probably every scientist you know will be like, yes, I agree, race is a social construct. This is what happened in my Black subjects. This is what happened in my white subjects, right? There's kind of a problematic construction there where we're not thinking about, we're like, yes, I agree with that statement, yet here I am using racial categories, right, to think about my subjects. So race is not based in biological reality. There's no biological coherence to the idea of race. It is a social construct, but race is a social reality. Right? The harmful effects of historical and contemporary racism have real impacts on lives, bodies, and environments. So bodies have been affected. Bodies of certain races have been affected by racism. This is where it starts to get really tangled up and hard to parse out because you're like, but this is what, like Black maternal health, terrible outcomes. It's not because of biology though, right? It's not because Blackness, that Black vaginas are different than white vaginas. Tell that to Jacques Rebel. But anyway, I'll talk about it later. So I worked on this paper with a couple of human microbial ecologists, a human evolutionary biologist, and a geographer, a human geographer. The paper was called Chasing Ghosts. So we talked about race as a ghost variable. So race is often used as a proxy in science when you actually mean other things, right? You're actually trying to get at other things, but race is a really easy way to group people. So we've seen a lot of microbiome papers that use categories of developed or underdeveloped microbiomes, right? Microbiomes by ethnicity, genetic ancestry, the term hunter-gatherer, which is, as an anthropologist, a strange, that's like ancient humans. That's not something that happens in 2023, right? You know, we talk about traditional or uncontacted microbiomes. Again, like all sorts of things where really, anthropologists should be involved in these studies to help you get your language better. So really what we might mean is that microbiomes are different because people have access to clean water or not, right? Because they have consumed low fiber processed foods, because they have toxic exposures, right? So these are the actual things that are happening that affect microbiomes, right? Those are the bigger structural things that cause the sort of middle column things. but we just kind of label everything as race. So what microbiome researchers might mean or might want to know about instead of race are things like diet, environmental exposures, access to healthcare, clean water and sanitation, stress from systemic racism. All those things are going to have an impact on your microbes, right? And Jordan and I were talking earlier about the fact, like in the Jacques Ravel study, makes very racialized claims about what microbes are in different races of people's vaginas, right? That's not scientifically accurate because race isn't a real biological category. So maybe think instead about what are the sexual practices and partners of those people? What are their diets like? What are their environments like? And then we can look in a more nuanced way about how those things affect microbes, right? Does that make sense? So in this paper, in order to build an anti-racist microbiome science, there are some interventions that have to happen institutionally, better representation in who teaches, does, and evaluates science. This is a big ask, I know, but reforming structures of like grant making, jobs, publications. If you are a paper reviewer and you get a paper that talks about racial categories and people's microbiomes, ask the author, why are you using race here? What purpose does it serve in your study? And what do you really want to know about these people? Because it's actually kind of just lazy to use race as a categorization of people. Because it's not real science, honestly, right? There's an essential transdisciplinarity to anti-racist future for microbiome, is that people like me who are experts in social science, public health, epidemiology, who have different kinds of knowledge, can bring something to your study from the beginning that might help you account for the social determinants of human and environmental health, right? And that no one discipline can do these things, right? You're all trained in a very specialized, highly technical way. And that's great. And that is what you should do. You shouldn't be responsible for all of the ethical, social stuff too, right? It's not your field of expertise. And I think it's too much to ask any one discipline to do. That's why we have to work together. Right? And finally, thinking about embedding in research design and community desires, demands, and needs. Asking people you're going to sample for their microbes, what are their health priorities? What do they care about? What is important to them? Do they give a shit about what you're studying? And is there going to be any benefit sharing? What do they get out of it? There has to be more parity in those relationships, right? Because we seek out samples from specific populations that really don't see any direct benefit from the science, right? And may not really care about the furthering of scientific knowledge. And I'm happy to send this slideshow, around. So these are a couple of papers. Where is on this one? So these papers that I've worked on recently is sort of addressing this issue and sort of where I see my future research going. And these are just my thank yous. But now I'm actually really done. That's all the slides I have. So thank you.
As per tradition, we like to take our first question from a trainee. Trainees want to start us off?
Yeah, I'm really interested in how you kind of went from a very highly like applied anthropology space into working with like microbiome scientists, like sort of that leap and how you maybe overcame like maybe some of the like learning challenges and things and like collaborations.
Mm-hmm. So I was really green. I was just a PhD student. I knew nothing about microbiome science. I know Jeff Gordon was the father of the microbiome. I certainly had no idea that like he was such a prominent and important person in this field, just because he was so generous and magnanimous and really welcomed me into that. So when I first went to the lab, I thought I was going to be studying the scientists. I never intended to do that kind of anthropology, to go to Bangladesh or do any of that. And that was the work I ended up doing for the lab. And so there was a lot of shifting and figuring stuff out over the course of a long period of time. And the book is really a lot, mostly about our collaboration and how difficult it was. And like, spoiler alert, in the end, how it kind of failed. Sorry to ruin the end of the book for you, but not like it was a terrible falling out or anything, but it was so, there were gaps that couldn't be crossed, right? But also how it has informed everything that I want to do now. But I think, maybe it was my naivete was beneficial to me. I didn't know what was happening and I was trying to figure it out. But, in hindsight, now that I'm a much older person and scholar. So here's the thing, the other thing about interdisciplinarity. Cultural anthropologists really don't like me very much. I've had submitted grants to Wintergun, which is the anthropology funding, institution, where peer reviewers have said, this is not anthropology. It's way too sciencey. You're way too in the science. And then, and anthropology faculty that have told me, like, you've crossed the line here. You've lost your critical edge, all of these sorts of things. On the other hand, like many of the junior scientists in the lab were sort of afraid of me. They were like, what are you going to write about? They didn't want to talk to me. And they were like, you don't even understand what your work has to do with our work. So you end up being in this sort of liminal middle place sometimes in interdisciplinarity where you don't have a home. So trying to figure that out has taken me a really long time too. But honestly, with this book, like I've been invited to many more microbiology departments than I've been invited to one anthropology department. But I think that says something. And it's sad because I think also my work is really saying anthropology has to change. Science needs to change, no doubt about it. Anthropology also needs to change. Anthropologists just write books for other anthropologists, and who cares? Like, I don't know interest. I just don't have any interest in doing that. Like, it does nothing in the world. Maybe there's a value to it, sure, but I am not interested in it. So we also need to, anthropologists don't want to account for the body, the biological material. They're like, let's think about the social and all these concepts. But we do have bodies that are being felt. So I hope that answers your question. Yeah.
So I had a question about the ethics around that. You mentioned that there needs to be more parity between the benefits seen by the people being sampled as opposed to the researchers. And so, I think and I know you're kind of coming at that from the point of view of like sampling people from Bangladesh or specific research populations. But, I do some research and I sample from people in the state college area. Yeah. And on my IRB form, what are the benefits to the subjects? I always put none, because we are not directly trying to benefit them. It's more of a broad scientific endeavor that would hopefully indirectly benefit them. And in some cases, maybe you can think of that for these other populations as well. So I'm just wondering what's enough of that benefit that's being conveyed to the populations.
There's been a disproportionate focus on what the science gets, right? So even your population here, I'm sure have some health priorities they would like addressed, right? And why should they give you their microbes? Honestly, why should they?
I mean, they're volunteering to do so. No one is. Yes,
absolutely. No, absolutely. But why should there be this disparity in who benefits and whose work is more, or lives are more important, right? So asking people, I think, is the first, like having a community engagement where you're like, what matters to these people? And if you want to know about microbiomes, here's where we will educate you also in the work we're doing and how it might matter to you, right? We can't keep science over here in this bubble that's separate from like all the mess of society and be like, yes, we're advancing knowledge. I don't think that cuts it anymore, honestly. I'm sure there are a lot of people there that disagree with me, but it's not good enough.
So when thinking a lot about race in the microbiome, which is an area we're involved in, there's definitely some tension that there's communities that think we should abandon the word race completely. And then there's the issues of having representation in the microbiome sciences when the data sets are 90% white European biased, right? And then there's also the aspects of health disparities and thinking about how do we connect the biology of the microbiome to health disparities that might be solvable? And so I feel like there's two messages. One is abandon at all costs because it's useless, which is somewhat coming from your presentation. Two, no, there is value of thinking about race through the lens of racism, health disparities, and diversity increasing in the microbiome sciences. And so we've got to find a middle ground, in my opinion, about how to not incriminate the use of race, but to actually say what is good about the data we have now with respect to race, and then where the field needs to go from there, which I fully agree with, should be less race categorized. But currently our IRBs, our clinical cohorts, all that moves through the data of race without all the metadata. So in the end, we have what we have now, but we can have a better future. And I think we can all agree on that message than maybe incriminating investigators who have been in this area of race and over assuming that they're doing something, you know, that might be the best they can do at that moment because the data isn't there right now.
Yeah, and I think the problem is using the category of race uncritically. Just slotting people in by what? Self-identification? That's a problem from the start. That already is like, do you see how like unscientific that is? You're asking people what race they are on a form and then saying that your results are dependent on those completely kind of arbitrary categories. So
But
yes,
some people are very proud to be identified in those categories too.
yes, but it may have nothing to do with their microbes,
That's
right?
true,
So,
but
yes.
when asked with self-identity, some people are very proud and want representation in the biological sciences.
Yes, and I am not a proponent for throwing out race. We can't, that doesn't do anything either, right? Absolutely agree there. But you can't, I mean, no, in the Chasing Ghost paper, we looked at PubMed, all the microbiome papers that have been published since 2005, all the ones that mentioned race. It was like, 2000 or something.
Yeah.
And like one talked about racism. That's not acceptable. If you're going to talk about race, you have to talk about racism because the impacts on bodies are because of racism, not because biological difference in race,
Definitely,
right?
definitely.
So yeah, so maybe we do still need to use race, but also additively, right? And like you're saying, collect all these other metadata, all this other metadata to also think about lifestyles and all these other things. It's so complicated, but I agree, like, and these NIH and NSF mandates of like increasing diversity of research pools and who your clinical subjects are and research subjects is not a good solution. Now you all just like have to have more representation, which doesn't change anything or the structures that cause problems in the first place. So now it's just a mandate that is sort of meaningless, honestly, right? It's not helping fix the problems. But I keep like harping on Chakra Bell, but like there's no reason that study needs to talk about race. It really doesn't. Categorize those people in a different way, right? Like I know someone, Johns Hopkins, who replicated that study with undergraduate students of different races, and they all have the same microbiomes. So like it has, like, it's not, it would be easy for someone to disprove that study, but that just had become a foundational study that now people hold on. So, do you know what I mean? Like race has no place in that study, honestly. It doesn't need to be, the subjects don't need to be categorized that way. So absolutely, there's so much work. Yeah.
If what we're doing now is not the right way to go about this, what is your suggestion?
In terms of categorizing people by race?
Yeah. And are you capturing that biologically relevant information into our data collection? How should we do it?
So I think that if you're thinking about using race, what is it really a proxy for, right? And get more precise about what you actually want to know about people's lives. And unfortunately, yes, then it becomes complicated. The reason they didn't talk about sexual health or partners in their Revell studies because it's super hard to collect that. Like, how are you going to collect people's like sexual history data and like continuing sexual practices and know that it's accurate? That's tough. So part of it is maybe having like public health people and like other people who are experts in sexual health as part of your study to help you collect that data. But that will tell you more about why people have certain microbes in their vaginas instead of just saying it's because they're of history, right? Like that will help you do your science better. But you have to ask other experts to help you, I think, and also just think the question differently from the start. right? I don't know if that's a useful answer.
One of the things that we love about your gross variables paper is actually this fact that you bring up industrialization is this term that is just used all over in the microbiome field. We have industrialized microbiomes and we have not. It's the big thing that splits globally. You know, you do big global comparisons, that's what you see. And it drives me nuts because I'm like, what is industrialization?
Yeah.
And also industrialization was a process that happened in the 17 and 1800s. And are we really, is that what we're looking at? Or are we looking at things that have actually happened very recently in the last 50 years? Like globalization or modernization, that's a horrible term as well. And so I guess, do you have comments on that and about, because again, it's kind of this idea of race. Like if we're categorizing people into industrialized or non-industrialized, we lose the ability to actually find out what's going on and what's driving, you know, microbiome differences. Do you have any comments on that industrialization sort of term?
I mean, you're right. It's just like race. There's no agreed upon standardized definition of it, but then there just is an expectation that you know what you mean by industrialized. So, I mean, I still think this has to come from an institutional level where funders and journals are like, define that word. Like they have to be like, no, you have to, you want this paper published, you want this funded. What do you mean by this? And that's maybe that, I don't know if that changes harder or, you know, easier to do at that level than on an individual scientist level. But yeah, I think if we're gonna be scientists, let's do it right. Like what, you can't just say industrialized. What does that mean?
Yeah.
Yeah.
I thank you so much for your talk. It was very provocative and I think all the good ways. I'm curious as you approach your subjects and you're in the field, I am curious what sorts of methods you use for acquiring information. I mean, obviously photography, are you doing audio journals? You're writing clearly, but like, I'm just curious what your approach is.
I mean, ethnography is like, can be many different things, right? Like semi-structured and informal interviews is most of it, right? Like a lot of it was just me sitting in people's houses in Bangladesh and like people were coming and going. I mean, I only had informed consent from the homeowners and like 65 other people would circulate through the house. But, you know, and doing more formal interviews, but I'm like, participant observation is a methodology of anthropology, which means you're just like in a place while people are doing things. And of course we recognize like observer bias and the fact that like one weird white person sitting in this house was certainly affecting people's behavior. That was not, there's no question about that. But I still had a more granular view of what was happening than the Gordon Lab, right? So I guess just like the Bangladeshi diets, it's like good enough data where I can sort of see what's going on. But yeah, there's lots of different methods, but that's primarily what I use was really just.
Do you record, audio record, or do you?
Yes, because I don't speak Bengali, I had a translator.
Yeah.
So it was, audio recorded, and transcript.
Okay, with that, we are out of time for today. If you guys have any burning questions, you're welcome to stay after and ask them. As always, there's a lunch for trainees afterwards.
Fantastic.
Thank you.